Hello, everybody. Wow, you guys are super excited to be here. I can see that, and thank you so much for your enthusiasm. I see a lot of young people here, so I want you to think about this like if you were in class and your teacher brings in a very wonderful guest speaker, right? And how would you act if you had a guest speaker in your class taking the time to come to your class and speak to you? You would be wonderful. So let's keep that in mind, and let's keep that in mind in the chat. Let's keep it in mind in the chat that you should stay on, stay on target. Let's keep talking about what Dr. Wilson will be talking about. I will be minding the chat, and don't be offended if I delete your chats if it seems a little bit out of context, okay? But anyway, my name is Bertha Vasquez, and I am a science teacher in Miami-Dade County. I'm also the director of this wonderful program called the Teacher Institute for Evolutionary Science. We give teachers resources to teach evolution in their classrooms, and we also provide wonderful opportunities like this for teachers and students to chat with real life scientists. And I was just telling our guest that at the beginning of the school year, I often ask kids to draw me a scientist. And I always get a crazy old guy with crazy hair trying to blow up the world with chemicals. And it makes me really sad that that's what kids and adults think of when they think of a scientist. You should be thinking about the people who made your cell phones, and the people who brought you television, and the people who keep you alive with medicine. And you should be thinking of people like Dr. Alex Wilson. So I'm going to introduce her now. Dr. Alex Wilson is our guest today, and I'm very excited to have her with us. She's an evolutionary biologist, and she studies symbiotic relationships. She will tell us a little bit about her research with aphids. Yes, Sunak, that is an aphid. And she's going to take your questions about what life is like for research scientists at a major university. Because maybe some of you guys are thinking about a career in science, and that would make us both, Dr. Wilson and I think, very happy. So Dr. Wilson, it's all yours.
Thank you so much, Bertha. It's really great to be here tonight, and welcome to everyone. It's a little strange to be talking to you guys because I'm just talking to the little green dot on the screen right here. I might have sort of defied Bertha's description because my COVID hair is pretty much looking scientisty these days, as my kids were laughing about last night when I was talking about doing this. So I may be embodying a scientist more today than I was 6 months ago.
And I should say, Dr. Wilson, if you don't mind me interrupting you, I will take your questions for Dr. Wilson in the Ask a Question section. It's at the bottom of your screen along the black taskbar. That's where you put your questions as she goes along. Thank you. Sorry.
That's okay, because I can't see them, guys. I can only see my screen right now. So Bertha's going to feed your questions to me as they come. And if you have questions along the way, please interrupt. And so that's fine to interrupt. And in fact, I encourage you guys to interrupt because it'll help me do a better job of making sure that I'm explaining things clearly. So yes, what I work on, what we study in my group is are insects that feed on plant sap. So and this picture here is of a female pea aphid, and that's PEA. They feed on pea plants. And then the small ones, if I move my mouse, can you see that? Can you guys see me moving my mouse?
No, I do not. But maybe, we still see the slides along the side. Maybe if you enlarge it and we just see your screen. But I don't think so.
It's full screen right now. I don't know if I can.
Okay, never mind. No, we do not see your mouse.
No, okay, never mind. Okay, I'm pointing at the small aphids. So they're the babies from this mother aphid here. So aphids are these small sap-sucking insects that feed on plants. And that's what we study. And we actually study some other insects that also feed on plant sap as well. But the first question I wanted to hit you guys up with, which I guess is a little hard, but maybe you can answer in the chat, is who knows what symbiosis is?
If you guys want to answer what is symbiosis in the chat, no peeking on Wikipedia or anything. It's okay to be wrong.
Totally okay.
You
to be
know,
wrong.
yeah, and science, being wrong means you get to learn something new. So we're cool with you being wrong. What is symbiosis? Oh, we got some answers, Dr. Wilson.
Okay.
Felix writes, 2 organisms that do things for each other for mutual benefit. Harina Garish says, when 2 organisms work together to survive. Oh my goodness. A partnership between organisms. Oh my goodness. You should see all the answers. 2 organisms that do things for each other. Relationship between 2 organisms where both benefit. The contact of 2 organisms, 2 or more organisms coexisting to help each other survive.
Good job, guys.
2 organisms. Yes. Oh my God. I think we have like 30 answers. It just exploded.
That's fabulous. Well done. So yeah, symbiosis is derived from ancient Greek, and the if we break the word down, the breakdown of it is together and living. So this idea of 2 organisms living together, and it can take different forms. So it can embed and sort of encompass things like parasitism. Who's had ringworm? Raise your hands. Raise your hands. We had it in our house a couple of years ago. Who's had head lice? That's parasitism. Commensalisms are just kind of the coexisting, say perhaps more like anemones and clownfish. And mutualisms are the relationships where they're meant to be mutually beneficial in both ways, that both partners derive a clear benefit out of it. And these are the ones that a lot of people have started working on in the last 10 years. And these mutualistic relationships are the ones that we work on in my group. So if we go back to thinking about insects that feed on plant sap. So there's 2 types of plant sap, right? You guys, what are the 2 types of plant sap? One goes up and one goes down. What are they?
Oh, wow. Well, I know that that's taught later on in the year. So if any of my students are in here, they don't know that yet.
We've got teachers there though, right? What about my kids? Are they there? Do they know the answer to this? All right. Well, we've got phloem sap, which is the nutritional part, and xylem sap, which I haven't written the name of that on this slide. It starts with an X, Xylem. And the insects that I mostly work on are the ones that feed on phloem. And so phloem is something that moves the nutrients around the plant. And if we look at this graph, but the thing about this, the phloem sap, is that it's a diet that's super rich in sugar. So it's essentially like junk food. There's so much sugar in it. And it has very few essential, very low concentrations of amino acids. Now, what are amino acids? I should tell you about that. So amino acids are the building blocks of proteins, and proteins are all the molecules that make up our body. So we have proteins that are enzymes, we have proteins that make muscle, we have proteins that do structural things. Without proteins, life basically wouldn't exist as we know it.
So let me, we're only seeing your beginner slide. We need could you click on the one with the graph that you're talking about? We're all seeing the wonderful beginner slide with the UM and the NCF.
Really?
Yeah.
How do I?
It should be just like a regular screen.
Yeah, it is a regular screen, but you're not seeing me go from slide to slide. What is symbiosis?
No, I didn't realize you were moving forward until you mentioned a graph.
Moving. I've even on says go to slide. Do you see it now?
No, we are seeing. You know what I'm seeing? There it is. There it is. We got it.
Now you see it. Okay.
Yep.
Well, I guess we'll just have to do it like this.
Yeah.
We'll make that really small, and we'll get rid of this. Okay.
There you go. Perfect.
So here we had this one. What is symbiosis? And then we had this one. And now, okay. So here we go. Here's this one. This is important. Okay. Oh, well, now we got everything. Okay. Here we go again. So aphids feed on plant phloem sap, and phloem sap is unbalanced. It's very low in essential amino acids. So these, as I said, are the building blocks of proteins. And there's actually 20 different amino acids in proteins. And so the 20 are listed. Can you see the graph here?
Yes, we can.
Okay. Along the x-axis, you see they're listed here in alphabetical order, all 20 protein building amino acids. And if we say that if we add up the amount of all of these, the molarity adds up to 100%. It's a little bit of an unusual way to represent it, but anyway. So if we look at all the amino acids, and this is the amino acid analysis of a plant called alfalfa, which is a host plant for the pea aphid. If we look at them and then we measure all the amino acids, most of the amino acid representation in alfalfa phloem sap is in the one form of this amino acid called asparagine, and the 3-letter code for that is ASN, asparagine. And you notice too that I have there's 2 different colored bars on this histogram. There's green, which represents the non-essential amino acids, and there's pink, which represents the essential amino acids. Now, the key difference between essential and non-essential is basically defined by what we as animals can make. So as animals, we can make the non-essential amino acids. We have the genes in our genome, the metabolic capacity to make these non-essential amino acids, but we do not have the metabolic capacity. We do not have genes in our genome to make the essential amino acids. And so we have to acquire, we need those essential amino acids because they're needed to make our proteins. But in order to acquire them, we have to eat them. We basically acquire them through our diet. And so here's the thing, is that this is what an amino acid profile looks like. And the thing about aphids is their mouth parts are such that they're like a straw, and they can only feed on phloem. They can't chew anything. They can't suck up cell contents. All they do is they navigate between the plant cells, find the phloem, and plug into a single sieve element in the phloem. And then actually by positive pressure, because the phloem's under positive pressure, the phloem just flows into their bodies. And so this is what phloem sap looks like in alfalfa. And then here's the problem. Here's the problem that aphids are faced with. They're faced with this challenge. So if we break this histogram down and sort of summarize it into this pie chart, 2 of the amino acids, asparagine and aspartate, represent, what's this, like 80% of the concentrate of the phloem sap free amino acids. And glutamine and glutamate represent, what, about 5%? And the essential amino acids represent here only about, what, 10% of the phloem sap. But this is what an aphid looks like. If we do an amino acid analysis on an aphid, the breakdown composition of the amino acids looks like this. So this pie chart is very different to this pie chart. And what happens in between eating and becoming aphid is metabolism. And in particular, the metabolism that we care about when we think about amino acids is amino acid metabolism. I wasn't going to do this here, but I want to show you guys this thing because it's so amazing. So you guys all know how to use Google. And if you go to Google, can you see my Google screen now? Can you see that? Can you see?
No, we're seeing your PowerPoint.
Okay, so now I have to stop sharing that and go share this other thing. Sorry, how do I do that?
Go to share screen, and you should, I've never, go to share screen.
Can you see Google now, guys?
Yes.
Okay, so here we go, Google, and we put in KEGG, keg, like not a beer keg, a keg. This stands for Kyoto Encyclopedia of Genes and Genomes, and this is something you can do, and you can click here, but this one here, we want to go to KEGG pathways, and here we have all these pathway maps. Metabolism, number 1, global overview. Remember how to get here, because this is like truly amazing. And then we go this top one, number 1, 0, metabolic pathways. Look at this. This is like a map of all of metabolism.
Whoa.
Isn't that crazy? And so
please explain what that means.
Well, this is like everything that happens inside our bodies and inside animals. And actually it's more than what happens inside our bodies, because as animals, we can't do all of this metabolism. Some of this metabolism only plants can do. Some parts, bacteria and plants can do. Some part fungi and bacteria can do. And some parts like only we as animals can do. If you really get into playing around with this site, you can actually pick a species, and you can see actually here, I think we can go here and we can say this is what humans can do. And it's gonna, hopefully it works. So from all of that metabolism that is known to mankind, this is what humans can do of that. the green stuff. So this is kind of a fun thing to look at. And one thing, so I'm talking about amino acids. Amino acid metabolism is described by all the yellow parts, the yellow here. And so we can actually scroll down here and we can go and we say we want to just look at amino acid metabolism. We can click on this and it just highlights. See how it's made that pink color, the bolder pink color? So that's amino acid metabolism. And this here is going to come up later. This circle here, this is the citric acid cycle. No, wait. Yeah, the citric acid cycle. We can go, oh, it's the wrong way. Got to go the other way. Oh, it's not going to move. All right, anyway, we can, so remember that keg. And I can put something in the comments, so I'll give something to Bertha and she can share it later. So now I'm going to go back to my presentation, but I just wanted to share, that thing is so cool that it's the sort of thing you just can look at and explore and just have your mind be like completely blown away. So now I have to go back to sharing the other screen, application window. No, not that one, this one. Okay, here we go. So metabolism, all of that stuff, and the amino acid bit, the bit I highlighted at the end, happens in here, and we go from having this amino acid profile of the alfalfa phloem sap to the whole body. Okay, so the question that I'm really interested in asking, and this is the thing that kind of makes me get out of bed in the morning and keeps me up. Actually, I tend to, these days to wake up in the early hours of the morning and lay there thinking about this. Is how do you go from being two separate organisms to being in a symbiosis that is so intimate that one partner lives inside the cells of the other partner? So the thing I didn't tell you here yet, the thing that how aphids manage to feed on a diet that's so unbalanced and looks so different to what their body type looks like is that what they did was 160 million years ago, they entered into a symbiotic relationship with a bacterium by the name of Buchnera aphidicola. And this series here of incredible images was taken by my former student, who is now my colleague. She's a Taiwanese scientist by the name of Xiaoling Liu. She lives now back in Taiwan. And she took these amazing images here at the University of Miami. And what you're seeing in the red stain, that's marking actin. So this is a structural protein that helps cells have form. And the blue, oopsie, we're flying through. The blue, what you're seeing in blue is a stain that stains DNA. Okay, so that's the you know, it's in the nucleus of the cells or in the bacteria. The bacteria, of course, don't have a nucleus, but it's also inside bacteria. And then the green here are the Buchnera. So that's actually a trick. She's actually pseudo-colored that. So they were actually stained with DAPI as well, with the DNA stain. But in order to make this image, she went and she made artificially made the Buchnera green. And so what you're seeing here is this is a very early embryo, a very early aphid embryo. And this is the stage where the Buchnera invade from the maternal cells that the Buchnera are living in into the developing embryo. And the incredible thing is that at this point in development, there are more bacterial cells in that little embryonic aphid than there are aphid cells. And then later in development, this process called cellularization happens, and the Buchnera get put into packed into these insect cells called bacteriocytes. And so that's what you're seeing here. You can't see. Can you see my mouse now? You can see it?
Yes.
Okay,
yes.
Good. So here are the bacteriocyte cells. And then later in development, so this is a pretty late stage embryo, they actually form into this organ called the bacterium. And so it forms down two sides of the body. And it is actually connected. It's just that when you take an image like this, you're just slicing through the organism. And so you're only seeing it in one plane. So if you go shallower or deeper, you're going to see that this is actually connected as a single organ. And so the question is so for 160 million years, these bacteria have been living inside aphids. And the evolution in the bacteria has happened in a way that their genomes have lost so many genes that they can no longer make their cell walls. And so they can no longer live outside the host. And equally, if you use antibiotics and you kill those bacteria, an aphid does not reproduce. So the relationship is so intimate and so dependent that the line between being sort of 1 versus 2 has almost disappeared. And the thing that I'm really interested in is like, how does that happen? How does that work? What are the molecular and cellular processes that result in having this functioning whole organism? And I'm going to tell you about one amazing thing that's happened in the process of this evolution. Here's just another picture. I don't think we need to spend too much time on it now, but it's another picture of the invasion. So this is just embryos and cells stained with DAPI, but the color that the scientist has chosen to represent it with is just a different color. Just, you know, artificially in like Photoshop or Illustrator, you can choose what color you're going to represent it as. So again, it's staining DNA. Again, you see the Buchnera invading. You see the organ forming, and then this is a bacterium that's been dissected out of a young aphid, and you see all these bacteriocyte cells, these big round cells that are full of Buchnera. So this big bright spot in the middle is the nucleus of the insect cell, and all those little tiny dots, they're individual Buchnera cells. And there's actually a pretty special arrangement. So each individual Buchnera cell is surrounded by a host membrane, which in this system we call the symbiosomal membrane. But actually there are pathogenic bacteria, things like chlamydia and tuberculosis, that also have an intracellular lifestyle. And when they invade cells in the host, they also end up with this host-derived membrane surrounding them. So that seems to be a particular feature of situations where you have a bacterium that lives inside a host eukaryotic cell. And so the thing is, there's lots of insects that have these bacterial symbionts and are dependent on them for helping with nutrition. And so these things here, this is a Milibug, this is a whitefly, this is some kind of a psyllid, and this is aphid. So these are all plant sap feeders. But here we have, what is this? This is a cockroach. So they also have symbionts, ants, and then here we have tsetse fly and the human body louse. So both of these are blood feeders. And so there's very curious things that have happened during evolution is that what these microbes do is that they supplement things that are short in the organism's diet. So for animals that feed on plant sap, that's essential amino acids because they're at very low concentrations in the diet. And for animals that feed on blood, the thing that these symbionts typically provide are B vitamins because blood is actually very low in B vitamins. It has lots of amino acids because it's full of protein, right? But so amino acids not an issue, but vitamins are an issue. And so one thing that we discovered when we sequenced the genome of the aphid was that the aphids lost some genes for some essential amino acid biosynthesis. So, or actually arginine is kind of the essential, non-essential. As adults, we make enough arginine as a byproduct of the urea cycle here, but as juveniles, so you guys actually require a dietary supply. You don't get enough, you don't make enough yourself, so you have to eat, attain it in your diet. And it was very surprising that aphids had lost. So this representation, this is the metabolism. So the circles represent the metabolites, and then in between is the sort of arrows are representing the enzymes that are mediating these reactions. And the ones that are shown in gray, this enzyme that does this black, this one in the middle is retained, and the ones shown in gray have been lost in the aphid genome. Well, it turns out the Buchnera, the symbiont, is able to help out. So the arrows in this picture, so these are amino acids. This is glutamate, and then glutamine, and then glutamate, and then arginine down the bottom. So remember, this is arginine here. And so the aphids lost the ability to make arginine. These red arrows, these are reactions that the aphid can do. And these blue arrows are reactions that the Buchnera, the endosymbiont, can do. And so the Buchnera is compensating for the aphid's loss of ability to make arginine. And so this was pretty extraordinary. So what we, and actually gets, I guess I actually didn't pick the most extraordinary one in aphid, but I have another coming in Milibug. And so this is another insect, the citrus Milibug. It's a pest of citrus. And they have these little tiny waxy things. I actually took some to class today to show the students because we were reading actually this paper that I'm going to show a figure from next. And they're about, 1 to 2 millimeters long, the adults. So they also have a bacterium, but it's a bit of a different structure to the one found in aphids. And this is even crazier. These guys actually have the endosymbiont has an endosymbiont. So this is a system where there's an insect cell. So this insect bacteriocyte cell shown in gray. Within the middle is the representation in yellow of the nucleus of the insect cell. And then around here, we have an endosymbiont called Tremblea that's shown in blue. And that endosymbiont, that bacterial endosymbiont has, shown in red, an endosymbiont called Marinella endobia. So it's like Russian dolls. You've got a bacterium living inside a bacterium, living inside an insect cell. And again, these things have been vertically transmitted from mother to progeny for millions and millions and millions of years. And in this system, and these are some of the scientists, this is a guy, John McCutcheon. He's at the University of Arizona now. This was his former student who was lead author on this paper, Philip Husnik, who's now a scientist in Japan, and Carol von Dolens at the University of Utah. And so Carol actually discovered this nested symbiosis. This is an electromicrograph from that initial work. And so here it's a little hard to learn to look at these, but this in the middle is the nucleus of the insect cell. And then these bundles around, that's the Tremblea, and the bright spots are the Marinella living inside. And so here in this panel that's marked G, the blue stain is a probe that's probing for the bacteria that's the Tremblea, the outside one, and the red stain is a probe that's probing for the Marinella, which is the endosymbiont of the Tremblea. And so this is an incredible nested symbiosis where you have this incredible arrangement. And then when we go and we look at the genomes of these organisms and we sequence the genes and we look, say, here at essential amino acid biosynthesis, I want to draw your attention to the key down here. So genes that are encoded in the Tremblea genome appear as blue dots. Genes that are encoded in the Marinella genome appear as red dots. Genes that are encoded in both Tremblea and Marinella are purple. That makes sense, right? Blue plus red equals purple. And then genes that are encoded in the insect genome are encoded as green dots. And so if we look, for example, at phenylalanine biosynthesis, we see that it requires genes from Tremblea and then, a couple of genes from Marinella. This one's kept in both genomes. Back to Tremblea, and then the final step in the pathway is completed by the host genome. So we have this incredible partnership where metabolism just cannot be completed without genes that are encoded in these three different genomes. It's mind-blowing. And the more and more insects that we look at that feed on plant sap like this, we just see the same pattern again and again and again, and often in exactly the same points in these pathways. And then in many cases, it's pretty clear that the situation is that these relationships, this interdependency, have evolved by convergent evolution. It's not a matter of shared ancestry. The same is true. This perhaps is not so interesting. There was another species here that's a comparison, but here I want to draw your attention to this bit down the bottom. There's three other colored dots, yellow and brown and orange. And these represent genes that have been horizontally transferred from bacteria. And so in this case, in the yellow, from alpha proteobacteria, from the brown, a gamma proteobacteria, from orange, from a Bacteroidetes. So from different lineages of bacteria have been transferred from bacterial genomes into the genome of the insect. And the amazing thing about this is peptidoglycan biosynthesis, that's got nothing to do with the insect. That's got to do with supporting the cell wall structure of the endosymbionts that have lost those genes in the genome. And so this incredible picture of gene loss and complementation of genomes, retention of genes to keep metabolic pathways, and then acquisition of genes from other unrelated bacteria to the genome of the host insect to support the cellular structure and the functioning of the endosymbiont. So just to sum up, like, this is not like the things I was talking about with the aphids. So that I already highlighted, I forgot to put Carol there too, but I highlighted already John and Carol and Philip. But these people here are people that have worked with me at the University of Miami, and these people in my lab, Dan Price, he's from Britain and he's back, he lives in Scotland now. Rebecca Duncan, she's American, she's living back in Atlanta now. Honglin Feng is from China. He was a PhD student in my lab, and right now he's a postdoc at Cornell University. Xiaoling Liu visited as a student. I mentioned her work, and she's from Taiwan. She's back in Taiwan. Chuck Lucci, my wonderful colleague from the medical school here at the University of Miami, he actually just retired. Maybe it's 2 years ago now. I miss him dearly, and he now lives in California. James Baker, he's American. He grew up in the Midwest, and he's a colleague here. He runs the microscopy core facility. And Atula Wickramanayake, who is the chair of our biology department here at UM, and he's originally from Sri Lanka. And without my kids answering, because I know they're out there, but like I'm not from the United States. We can have a guess right now as to where I'm from. You can put it in the chat. But anyway, I'm happy to take any questions.
Okay, I am unmuting my microphone, and I'm going to have my partner in crime, my Thai's partner in crime. I'm going to invite Kenny over. Wow, there's so many people in here. Let me find Kenny. He's going to ask the questions for us, if I can find him. If not, I know he was in here a second ago. I saw him. I'm going through this kind of fast. Oh, shoot. There's so many people in here. It gives you a chance to catch your breath.
It does.
You talked very quickly. That was...
Well, I was worried. Oh, but we're at half time now.
Yeah, no, we're fine. We're fine. I know he's here, but... Oh, there he is! Okay. So, Kenny is the question asker extraordinaire, and he's been invited to the screen. He's also a science teacher, and he's going to be the one asking the questions.
Hi, Kenny.
Hi, Kenny.
Hello. Hi, guys.
Hi.
So Dr. Alex Wilson, I played the second symbiosis video for 5 of my 6 classes today, where you say girls, 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 and I know that's Bertha's favorite part too.
No, except she doesn't say girls, 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 girls. She says gals, 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 something like that.
That was not so that there's a fabulous story that the 2 filmmakers who made that actually right here in this, like I just turned my chair around and I'm like literally the same chair, same spot, just facing the wrong way. When they filmed that, and they didn't, we had a lot of fun. I didn't know, but that was not scripted. They told me, okay, Alex, this is how it's going to go. This is the story. This is what we want to do. This is what we were thinking. So just do it. And yeah, we might have had to have a couple of takes of that one, but.
Okay, now I'm going to have to show that to my bio kids because they're like, what are they talking about? Yeah. But go ahead, Kenny. You have a...
Yeah. No, I mean, just for everyone who's not, if people don't know, if you look at my webpage, the University of Miami, if you Google Alex Wilson, University of Miami, and you go to research and then films, there's 4 short films about 6 minutes that were.
I'm going to do that. I'm going to put it in the chat.
Okay. Yeah. And they're kind of fun. I always feel awkward. The one that that second one about aphid biology is just amazing.
Yeah. I put it in the chat.
The photographs of like aphids being born and aphids pooping, and yeah, it's pretty cool.
Yeah. The whole class kind of gasps at the same time and laughs at the same time, and it's a great video. So we're gonna maybe stop talking about aphids just for a tiny bit because a lot of people want to know, do you remember what age you became interested in evolutionary biology, or what inspired you to pursue a career in evolutionary biology?
Yeah. So as far as the evolutionary part goes, I'm not really sure about that. I think that so as far as being a biologist, I think in many ways I was kind of always a biologist, and my earliest memories of being really interested in things biological really date back to being in my grandmother's garden with her. And I didn't actually realize how extraordinary it was that she knew the names of everything. she didn't know, she was not educated, she didn't go and have a higher education or anything, so she didn't know the species names or everything, but she knew the common names of everything. And so I grew up kind of knowing a lot of stuff that and I thought that was normal, but I don't think it was normal. And so I think I just always liked being outside. And then the tipping point for evolution with me was when I read The Beak of the Finch. You know this book?
Yeah, I have it in my classroom.
I have it right up here somewhere on my shelf. And for me, that was amazing because that book describes evolution in real time. And I think before that, it was just like dinosaurs and distant, like too hard to imagine how it actually happens. And then I read that book, and I was a PhD student actually at the time. So I sort of was, or I was actually not studying evolutionary biology so much, it was more population biology. But I think that, I really actually don't really know how you can be a biologist without being an evolutionary biologist. I think that like it's just a way of thinking.
Yeah.
Yeah.
And where did you get your PhD from?
Well, so someone guessed that I was from Austria. I'm not sure if that was a spelling mistake or a few letters missing. I am from Australia. Yeah, from Sydney, Australia. So I grew up. I was born in Sydney, but I moved when I was only 3 months old. My mother was British, so we moved to England, and then we moved back to Australia. And then I've been in the United States for 20 years, so I know my accent is really messed up now. But I did my undergraduate and PhD work in Australia at Macquarie University. That's the word that it's not Macquarie, it's Macquarie University in Sydney. And then I moved to the United States in the year 2000, early June 1st, I arrived. And I did a postdoc. So that's your like first job after you finish your PhD. And the first one I was at UC Davis, which is like the premier or one of the premier departments in the entire world for the study of evolutionary biology. And so that was really extraordinary. It was like, I had no idea. I just landed. I was like, wow, this place is amazing. And then I went to Tucson, Arizona, and did a postdoc there. And then I got the job here, which is pretty much as far away from Sydney, Australia as you can be on the globe.
Yeah.
So, yeah.
In all of your research, in your specific research, what was your wow factor? Like, what was your wow moment when you said, like, wow, this is really going to be impactful to science? Or maybe what was your first wow factor?
My first wow factor? The first one was probably the really first one was when I was doing my honors degree, which we don't have them here in the United States, but that's like a year for students who do very well academically in Australia. There's a 1-year research degree you can do on the end of your bachelor's degree. And so it's a full hands-on year of research. And I was working on rock wallabies, small kangaroos that hop around, a very good hopping across rocks, right? And the question was about what the species relationships were in the group. And there was some very old tissue samples in the freezer that we had trouble, like the freezers had broken many times over. And so the samples had, defrosted and being refrozen and defrosted and being refrozen. And if you want to get good quality DNA out of a tissue, that's a bad situation. And so the DNA gets very fragmented when it freezes and thaws. And that means that doing any sort of chemical amplification or PCR, polymerase chain reaction with it, is difficult. And so I think one of my first wows was like when I amplified degraded DNA and I got sequence out of it. So really, I think my first wows were just the being in the lab and like actually doing science. And back then we had to sequence DNA by hand using like radioisotope, and we would pour these big gels, and so it was very hands-on and very, it was less sort of magical where you sent the sample away and you got data back. It was much more that you were generating the data in real time. And so they're kind of small whales. It didn't change the world in any way. But for me, it was like just that sense of like, I can ask a question and it can be difficult to get the answer, but I can get through that. And yeah.
Aside from, so now a lot of people are very interested in these aphids, and aside from the symbiotic factor, is there another important reason to study this type of insect specifically?
I mean, there's lots of other cool biology. You know, that one of the big, they're big. Aphids are big economic, like crop pests, and so they're not pests for the direct damage they do because their mouth parts are so fine. They don't particularly cause any, like, it's not like a caterpillar, right? That, like, think about your milkweed. People all over the country, right? So it's not just like people in Miami where have we got lots of Miami people here? Yes.
We probably have a lot of Florida people too.
Yeah, so if people are familiar with, like, you know, how many people are trying to grow monarchs in their backyard, right? And putting milkweed out, and like you think one day you've got all this milkweed and the next day you go out and it's just sticks, right? So an insect like that just chomps the plant up. So aphids don't cause that type of damage, but what they do is they transmit plant viruses. And so everyone's really familiar with what viruses can do these days, right? And how much havoc it can wreak to it. So the big issue with aphids are that they transmit a lot of different plant viruses, and they feed on agricultural, you know, basically every agricultural crop you can think of, there's an aphid pest that specializes in it.
Are there a lot of different aphid species?
Yeah, there are thousands. You want to see some big books? There are thousands. But you know what? Down here in the tropics, not so much. So the aphids don't do well in the heat, and the reason they don't do well in the heat is their symbiont. So the Buchnera is not very thermostable, and if you remember, I said if you kill the Buchnera, they don't reproduce. So if it's too hot, it kills the Buchnera, and the aphids don't reproduce. So that's basically why we don't have that many aphids in hot places. But if you have got milkweed growing in your yard, you've probably seen those bright yellow aphids. They're little bugs that they'll be up around the flower buds, and they're literally bright yellow. They're the most beautiful bright yellow with black legs and siphuncular stuff. So, but there's another really, really cool biology that happens in aphids. I mean, if you watch the videos, one of the things you'll see is this telescoping of generations, and they can reproduce both sexually and asexually. But another thing, like I'm super excited about, I think is fascinating, although I've never worked on it, is that I mentioned that aphid diet is like super sugary, really high sugar concentrations, and the concentration of the sugar is actually so high that it creates an osmotic challenge for the aphid. So it's actually the sugar concentration is so great that it should essentially dehydrate the aphid. And that doesn't happen. The way they get around that is what they do is they cleave the glucose into fructose and sucrose, and then they make these long glucose chains. Sucrose into glucose. Then they make these long glucose polymers. And when you increase the concentration of sugar in the diet, the length of the molecule is longer, and they keep the osmolarity, the molarity of the diet down by making the molecules bigger. So it's a crazy situation. Really interesting biology.
Daniel wants to know what are they eating? What are the pea aphids eating? Only pea plants?
Oh, no. So there are some aphids that but pea aphids feed mostly on legumes. So in the lab, we keep them on fava plants. Sometimes it's spelled F-A-V-A, sometimes F-A-B-A. But you can, they also like to feed on alfalfa. And then there are some that are found on but mostly they feed on legumes. So most aphids have a very narrow, there are some that are generalists that feed on lots of things, and they're big crop pests, but many aphid species feed on only a very narrow range of plants. So as the plants have speciated, so the aphids have speciated.
And Isabella wants to know, what type of microscope are you using to study the aphids? And Lily wants to know, how do you dissect an aphid being so small?
Yeah, good question. So we use different types of microscopes to do the dissections. We use a dissecting microscope, and some of you guys might have a dissecting microscope in your at school, right? And then the images, the beautiful, colorful images, we use a microscope called a confocal microscope. And then the image of the black and white image of the Milibug nested symbiosis, that was taken using an electron microscope. So, but mostly, like most of the stuff we do in my lab, we're using a confocal microscope.
Okay. So we have to work on that, Bertha. Get better microscopes in the school.
You know, that's like $500,000 for a
microscope. We did we did electron microscopes in class today. Well, we studied what they are, what they look like, but we don't obviously have it. I have some good compound light microscopes, but that's about it.
But you guys can come by UM and we can show them to you.
Miranda wants to know, since the phloem is largely made up of proteins, do sap-feeding insects consume a similar amount of the other macromolecules?
I got to turn my pinging off. I'm sorry, that's my pinging. My son just realized he was too late, so he really...
He's joining.
No, I think everyone texts from different rooms in the house, right? It's ridiculous. So, sorry, what was the question again?
So since phloem is mostly proteins, do sap-feeding insects are they also eating carbohydrates and lipids and...
Oh, so sorry, I misled you. So it's mostly free amino acids. So it's not, actually it's mostly sugar. So sugar is a carbohydrate, right? So they getting the sugar, and then the amino acids are not in the form of proteins, they're actually in the form of free amino acids. So the building blocks of proteins. And the lipids, there's not so many lipids. So they're also kind of dependent on the Buchnera for making lipids. I didn't bring water in here. I'll go get some. But I also realized I didn't answer the question about dissecting aphids. The way we do that, excuse me, is we get like barbecue skewers. And we have these very, 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 we don't use the barbecue skewers. We have these very, very fine pins called minuta, and you don't, like, they're almost like hairs. And you stick one of them in the end of the barbecue skewer and put a blob of glue around it. And then you use those very fine pins to dissect the aphids under the microscope. And sometimes you have to make a bunch of them, and then you drop them on the counter to try and like make the pin bend a little, so it makes a little hook. And then you just find that you find one that you like dropped in just the right way, and it becomes your favorite.
In the videos, in the 4 symbiosis videos we were talking about, I don't know if you talk about it, but you have this really fun way of moving the aphids.
Oh yeah.
Can you?
I have one here.
Oh, good.
Wait, maybe let me see if I have one in my drawer here.
After you watch the video, I always...
We haven't been doing that because of coronavirus.
Yeah.
So I shut that down because so we used to use these days we're using paintbrushes. But we used to use a piece of like aquarium tubing with a pipette tip on the end to make a fine point, and then we put it in our mouth and like with a vacuum, like a mouth vacuum. But literally since COVID, we are not doing it that way.
I understand. Somebody else was asking. Yeah, Jay, I was wondering, has COVID-19 affected other aspects of your work?
Yeah, it's been really bad.
In the lab.
It's affected everybody, right? So when in March, when the shutdown happened, one thing that was a little bit nice was one thing that happens when you advance in your career as a scientist is you spend less and less and less time actually doing the science, and more of your it becomes something that your students do. And so with this shutdown, that meant that I started taking care of everything because I didn't want to be asking anyone to come in, and because we didn't know right at the beginning, would it be safe to come into the building? Would it be transmitted through the air conditioning and all this sort of thing? So that meant that I've been working with the insects a lot more. But unfortunately, it's also been really limited, right? Because I'm also a mom, and so I have 3 kids, and I've been at home with them. And it's not, and teaching, I had to switch to teaching online. As you guys know, it's challenge, there's a lot of things that became taking up more time, but we are getting back into the lab now. And but equally, one of my students, who was a PhD student, decided to leave the PhD program. So she's not doing research in the lab anymore. She became a science teacher. So so I don't know if she's out there tonight, but hi, if she. So it's been difficult, but and then when the undergrads came back, and we have some classes in person, most of their classes are online, but they're really keen to do research, but it's hard to do research when we can't be sort of close together showing. But I have one undergrad from Jamaica who came back, and he already knew how to do stuff, and so they're super excited. Like he's super excited. He's got an experiment he's setting up, I think, tomorrow. So yeah, it's definitely been hard, but I feel like things are starting to ramp up again now, and we're hitting a groove where we're getting to know what we can comfortably do, and people are starting to feel safe in the environment and stuff.
Christina wants to know, is symbiosis more common in vertebrates or invertebrates? And I think Berth and I talked about symbiosis meaning symbiosis or mutualism and parasitism.
Yeah, that's
so you can
question.
Yeah.
So when it comes to these intracellular systems, like I was talking about, tonight, that's much more common in invertebrates, and it's very common in insects. It's also found in other invertebrates like leeches and things like that. But we are covered in symbionts. And actually, I bought this. Do you guys know this book? I Contain Multitudes, Ed Yong. Fabulous book. Highly recommend it. I'm teaching a class right now where we started in the first 3 weeks. We read the whole book together. But if this is the sort of stuff that you're interested in, so this talks about, it talks about a lot of the things I talked about today, but it also talks a lot about like human microbiome. And so when we use the word microbiome, what we're talking about is a community, and our health critically depends on our microbiome, especially like gastrointestinal. We have microbes in our gut that we require to digest food. And in fact, like the human genome contains very few genes to digest complex carbohydrates, only about 35, whereas our bacteria that live in our gut contain hundreds and hundreds and hundreds of different genes that are able to digest complex carbohydrates. So symbiosis is important everywhere. And it's just the sort of different types of symbiosis, different types of complexity, but these sort of simple one-on-one systems tend to be found in invertebrates, and definitely not vertebrates.
Nushka wants to know, is the bacteria in aphids the same bacteria that we can find in the human gut?
No, absolutely not. So the one in aphids is the Buchnera that I was talking about, that is only found in aphids.
And somebody else wanted to know, are there other animals that have a deep bond with the Buchnera?
Nope,
but
aphids.
Just special to aphids.
Just aphids.
And Cindy wants to know, was the bacteria in the aphids, she wants to confirm that it evolved over time to not have cell walls.
Well, it's lost a lot of the genes it needs to make its cell wall, and especially with respect to peptidoglycan and some of the lipid biosynthesis. And so that's still, like, there are some people who've started to work on that more in Milibugs. Actually, John McCutcheon, the bold scientist that was there, he's doing that kind of stuff, but it's still not, like, there's still a lot to figure out. These are all really good questions, guys.
All right. I'm trying to get different types of questions.
Sure.
They want to. So Madeline wants to know: have the aphids evolved? Okay. If the aphids evolved, why did they lose the ability? I know it's not purposeful, but why did they lose the ability to metabolize certain amino acids?
So as animals, like all animals have, like, so inability to make essential amino acids was lost in the common ancestor of all animals. So that's nothing particular about aphids.
Yeah.
But the losing the arginine biosynthesis, it's not just aphids. Other insects have sort of done that as well. And I think, so that's one of the things we're working on in my lab, is I think that actually has to do with the mechanisms of integrating the endosymbiont and the host. So in terms of, so there's this metabolic pathway called mTOR, mechanistic target of rapamycin, and it functions to regulate sort of cell growth and like growth and cell death and all this sort of core processes. And I think that it might have to do with this sort of this integration. How do you go from being two separate things to a single functioning whole? But that's this is exactly the sort of work that we're trying to like get insight in on now.
Fenris wants to know if you could make one adaptation in aphids to help them evolve, so they could survive better. Yeah. What type of adaptation, what would you consider would be helpful for their survival, more helpful for their survival?
Well, okay, so think about global warming right now. Everything is experiencing a warmer planet, and one way to cure aphids of Buchnera is to expose them to a temperature of 30 degrees Celsius, which we experience very often in Miami, right? And so I guess a mutation that made them more heat tolerant would be something that would, ensure that they persist for another 160 million years.
Mm-hmm. To go back in time, Sean wants to know if the amino acids that aphids eat are so sugary, how did they survive before the Buchnera?
So amino acids and sugars are two different things, right? So amino acids are not sugars. Amino acids are nitrogen-containing compounds. And so, yeah, there's a conflation of two different concepts there. But I know I learned from Bertha lots of things in life, and one of the things I've learned is that it's pretty far along before you guys learn about like amino acids and all these types of molecules in the school curriculum. So I'm sorry to be talking about things that but you're all smart, I think just knowing that there's a lot that you don't know and a lot to be curious about is fine, and I think, one thing I know as a scientist and as an evolutionary biologist is I think we get very comfortable with not knowing the answer to things. Like it's not something to feel uncomfortable about, because it's like there's sort of three things. There's things that I think I said this actually in one of the movies. As I said, the sense of there are things that I know that are, like unknown and I know the answer to. There are things that are known but I don't know the answer. You guys might be in a lot of stuff like that right now. And then there are things that we just don't know. And as scientists, like we're not really doing a good job of doing what we do if we're working on things that we already know the answer to, right? So I joke, but it's not a joke, that I exist at the edge of my incompetence. So I'm essentially always working at the edge of what I know, which means that I'm working and daily faced with what I don't know. And that's a fun place to be. So
that's what teaching feels like these days.
Yeah.
Well,
so,
anyway.
So Dr. Alex Wilson, we're almost out of time. I just want to ask one more question. And I understand we see some people saying their questions were skipped, but we had so many great questions and we all want to go to bed and be ready for Friday. So this kind of will be the last question. How long have you been studying P. aphids? And would you ever change to a different insect?
Yeah, so that's a good question. So I've been studying aphids. I actually worked on aphids during my PhD, and I completed my PhD in the year 2000, and I started it in like 96 or something like that. So for more than 20 years now, a long time I've been working on aphids, not always asking the same questions. So the questions have changed a lot. I started working on the evolution of sex and understanding how genetic variation was patterned differently in sexual versus asexual lineages. But yeah, so I'm thinking about different systems, because what I really I've got interested in this sort of really mechanistic questions of how things actually work. And to really answer those questions well, you need to have systems where you can genetically manipulate the system. And the limitation, a really big limitation in working with aphids, is that you can't culture the bacteria outside the aphid, and there's no way to manipulate it genetically. I mean, essentially, it's so dependent. It's lost so many genes. It's so dependent on its host that it's kind of limited. So I have started flirting with the idea of starting to work in a system where I can maybe manipulate it using genetic tools like CRISPR and things like this more. I also started right outside my window, just there, we have some palm trees that we got from Montgomery Botanic Garden, because there's an aphid that feeds on palms. It doesn't look anything like an aphid. It looks more like a mealybug. But it has lost Buchnera, and it's replaced it with a yeast-like symbiont. So it's not even a bacterium. It's a eukaryote. And so I'm hoping that in that system, I might be able to get some insight into these mechanisms, so that the molecular and cellular mechanisms that integrate, are they the same when you lose Buchnera and get something else? Are these points the same? So yeah, good questions, guys. It's really, thank you for so many people coming.
Yes. Thank you, Dr. Wilson, for exposing us to all this.
Well, thanks for having me. It's been a pleasure.
Thank you.
Everyone stay well. And the biggest thing I have to share, this thing, do this, guys. Super. It's probably the single most useful thing we can all do to get out of this mess right now, right? Wear your masks everywhere. And as I say to all my lab, at the end of lab meeting every Friday, I say, and no kissing strangers. So
great advice.
Stay safe, everyone.
Thank you.
Bye.
Bye.
Bye.